So thank you for joining me, Dr. Cooper.
Thank you for inviting me.
What are your qualifications to speak on this subject?
I'm a veterinary pathologist with a Ph.D. who spent the last 5 years working on Ebola virus.
So, Ebola virus. What work have you done on Ebola virus?
We've done pathogenesis and treatment studies.
What exactly does that mean?
Pathogenesis is understanding how the virus causes disease. Treatment is trying to alleviate the symptoms of disease and prevent mortality.
How does one contract Ebola?
That's not currently known. It is suspected that it's through percutaneous exposures, including aerosols and mists, but it can also be through cuts and skin. The very first outbreak occurred at a couple hospitals, missionary hospitals in Sudan and Zaire, and so those were being spread by intramuscular and intravenous injections, which are obviously highly efficient but aren't very common in terms of outbreaks.
So, how does a virus like Ebola pop up? How does it?
There's an unknown reservoir, most likely a bat, and there is a large hunting and bushmeat trade in Africa. And whatever that reservoir is, it either directly spreads it to humans, where they do eat bats in some countries, or it has an intermediate host, such as some people suspect duikers or small primates, small monkeys might be an intermediate host, an amplifying host, such that the bushmeat hunter finds a sick animal, kills it, butchers it. In the process, if he cuts himself or already has any cuts on his hands, he exposes himself to the virus because the animals have a high titer of virus in their blood when they're sick, and from there it spreads to other people.
What exactly does the virus gain by spreading to other hosts?
Well, that's a philosophical, not a scientific question. Viruses are not themselves sentient, right?
Yeah.
So they don't actually gain anything, but they do increase their numbers.
How exactly do they increase their numbers?
Because when the virus gets into a new host, the first thing it does is start replicating.
How does it replicate?
It makes a copy of its RNA genome and then uses that as a template to make more RNA genome. It has an RNA-dependent RNA polymerase.
Now, once one does contract Ebola, what's the process of what happens from initial contraction to death?
There's a prodromal period where the person will be asymptomatic. The virus is already replicating. They will initially have some mild flu-like illness, aches, fever, body pains, nausea, vomiting, diarrhea are common. They will eventually become prostrate. They often have altered mentation where they can't think clearly. Usually at about 10 days after suspected exposure is when they start getting really ill. And from there, it's usually less than 2 weeks till death. There's multi-organ failure. The kidneys stop working properly. Cardiovascular system shuts down. There's hypotensive and hypovolemic shock. It's basically a cascading failure.
What strain of Ebola do you typically research?
It depends on the study, but we've used strains that were isolated from the original 1976 Yambuku outbreak from a young nurse named Mayinga. We've used strains from the more recent 2013 West Africa outbreak called Kikwit, and we've also used Makona strain, which was from an earlier outbreak.
What exactly are the major differences between those strains?
Nucleotides. There are minor genetic differences. They're all very well mapped out, but there are minor genetic differences in terms of missense and silent mutations in the various proteins that the virus makes, as well as mutations within non-coding elements such as gene enhancers.
So, Is there anything that the immune system can do to deal with Ebola?
Yes. So, depending on the outbreak, there is a mortality that ranges anywhere from 95 to 50%. So, obviously, there are people who get infected with the disease but are able to successfully fight it off. And that seems to be predominantly lymphocyte mediated, particularly T cells seem to be important. And there are also vaccines that are now available, as well as treatments, including monoclonal antibodies, which provide protection.
How exactly do they provide protection?
Well, they're synthetically created antibodies against the Ebola virus. So instead of waiting for your own body to produce antibodies, you can just receive them passively through the treatment.
I think fine. How exactly does Ebola spread through the body?
It spreads through the bloodstream, both as virus particles free within the blood and also within monocytes and macrophages that happen to be transported in the blood. The monocytes and macrophages are one of the major cell types which are infected by Ebola, in addition to a wide variety of other cell types, including the hepatocytes of the liver, endothelial cells of the vasculature, fibroblasts of all your connective tissue, synovial cells, microglia in the brain. There's a whole host of other cell types that are infected.
Is there a specific reason that it infects those cells?
Well, it uses a cell receptor, which is the Niemann-Pick cell-associated receptor, which is commonly expressed on a number of different cell types. So it's whatever cell type has the receptor that the virus can bind to that allows it to infect those cells.
Would you like to talk about a few of the studies you've done?
Well, we've done studies to, for instance, show that the virus, not just Ebola, but also the related filovirus Marburg virus, will infect the synovium of diarthritic joints, because arthritis is a common sequela of Ebola virus infection in people who survive, and joint pain is a common reported finding in people who have acute Ebola virus disease. We were curious to know whether or not that might be a target organ, and it turns out, in fact, it is. Nobody had looked at it in 40 years. Other studies that we've done have been looking at treatments, including monoclonal antibodies and monoclonal antibodies with small molecule therapeutics. We've done confirmation of virulence studies with new isolates just to make sure that they are, in fact, Ebola, and they do behave as we expect Ebola to behave. They don't cause, for instance, unusual disease or anything like that. We've done transmission studies looking at different routes of transmission, how efficiently it spreads.
So, obviously, Ebola didn't pop out of nowhere. So, have you done anything to find out, what diseases Ebola might have stemmed from or evolved from?
So, filoviruses are found in a lot of species that you wouldn't expect them to. And, for instance, fish have a whole range of filoviruses, but they don't seem to cause disease in fish. They're just sort of molecular parasites that hang around, which is why it sort of makes sense that there's an asymptomatic host, again, probably a bat, that does just fine with the virus, but once the bat spreads the virus to a non-host adapted host, such as a monkey or a person, it does poorly in that host. In other words, there have probably always been filoviruses, and there's probably always been Ebola virus, but because it doesn't cause the disease in its normal host, nobody ever noticed it.
What exactly makes you think that the reservoir species is a bat?
Well, it's known to be a bat for Marburg virus, which is the other related filovirus. It's the Rosetus aegyptius bat. Bats are also known to harbor a number of other viruses, such as Nipah, which is also a viral hemorrhagic fever. But, I mean, that's just supposition. Nobody's actually found the host. Lassa fever virus, which is another viral hemorrhagic fever, the host is the multimammate rat. So it may be a different species altogether. Could be a fish, as far as anyone knows.
What exactly is the survival rate of Ebola?
So it depends on the outbreak. It's been anywhere from 50% to 5% or even less than 1%. But a lot of those outbreaks were small numbers. I mean, there have been outbreaks as few as 3 people. That's kind of a small number. It's hard to get an accurate estimate of mortality. And there are lots of people walking around with strong antibodies to Ebola with no recollection of ever having been sick. So there's probably cases of either asymptomatic or mildly symptomatic infections. You have to remember that in sub-Saharan Africa, there's a lot of endemic fever-inducing diseases, things like malaria, typhoid. People might not notice the difference between a case of malaria and a mild case of Ebola.
So, approximately how many outbreaks of Ebola have happened?
I don't know the exact figure off the top of my head. The first outbreaks were paired outbreaks in Sudan and Zaire in 1976. There is either currently or just ended one in Central Africa. The largest one was in Western Africa, in Liberia and Guinea. I believe that was 2016 to 2018. And that was the biggest one by far in terms of number of people infected and number of people dead. But there have been over 20 recorded outbreaks. And the first outbreak of filovirus was Marburg virus in Marburg, Germany in 1967.
Have you ever contracted Ebola?
No, I have not.
Have you ever known someone who's contracted Ebola?
Yes.
Can you talk about it?
Sure. I work with a physician who was working on the outbreak in West Africa when he became ill. He does not know how he became infected. He was medevaced to the U.S. and received treatment, and he survived with some sequelae and some complications, but overall fairly healthy. We work with Ebola in a what's called a BSL-4 level facility, which means that we wear spacesuits under positive pressure. We take precautions to avoid any potential for infection during the course of our work.
Well, thanks for joining me, Dr. Cooper.
Thank you, and thank you for all of the excellent, well-written questions.